Hello and welcome to the fourth session. Today we are going to see the translation in prokaryotes. The process of synthesis of proteins, which is called a translation. In the case of prokaryotes, it occurs in the cytoplasm. Actually, transcription and translation are coupled in the case of prokaryotes because there is no spatial separation between the DNA or the genetic material and the cytoplasm. Because there is no such nucleus, etc. Whenever an mRNA is produced, that will be at the same time it will be transcribed. That is why both these processes are said to be occurring almost at the same time. So both transcription as well as translation are said to occur in the cytoplasm, and almost they are simultaneous processes. Soon after an mRNA is formed, that will be translated into that particular protein. So coming to the key points in the mechanism of translation, so here you can see that there is some difference in the headings that you see from the text. Not the main heading, you can see many subheadings in this. So I have designed the subheadings in such a way that each of these subheadings will guide you to the particular steps without without missing the points. So we'll see the steps. First one is charging the amino acid. It is nothing but the tRNAs which is in the tRNA pool should be combined with the particular the specific amino acid, that is what is called as charging of the amino acid, followed by the three steps, which is initiation, elongation and termination. Initiation, I have subdivided that into four steps. The first is bringing the 30th subunits to the initiation codon, initiation factors, then bringing the first tRNA into the ribosome. Then after that, assembly of the initiation complex, which is followed by elongation. First step is amino-acyl tRNA selection, peptide bond formation, translocation and release of the deacylated RNA, and the last one is termination. So these are the sections we are going to discuss. So before discussing the specific sections, we have to see three different terminologies. One is a codon. So a codon is nothing but the three-letter sequence that is seen in the mRNA, and these three letters correspond to a particular amino acid, or a stop codon or a start codon or something. So a codon is a sequence. It is just a sequence of three DNA or RNA nucleotides on the if it is RNA nucleotides, it is on the mRNA, which is corresponding to a particular amino acid, or a stop codon or a start codon, etc. And this is the particular signal during the protein synthesis. So a codon is the language that is read by the ribosome. Ribosome can read only codons, that is, the three-letter words can only be read by the ribosome. Next is anticodon. Anticodon is seen in the tRNA, which is also sequence of three RNA nucleotides, which is complementary to the codon in the mRNA. It is complementary. So anticodon is nothing but the complementary three nucleotides which is seen on the mRNA, which is complementary to the mRNA. That is what is called as anticodon. So codon is seen in the mRNA and anticodon is seen on the tRNA. And now an ORF, or an open reading frame. An open reading frame is seen in the mRNA. An open reading frame is nothing but it is a frame on which a protein is synthesized. It is a frame of a protein which begins with a start codon and ends in a stop codon. In the case of prokaryotes, you have a single start codon and three stop codons as shown there. So these are the three terminologies before starting the process of translation. After that, we have to see some details about the structure of ribosomes and their role in translation. As you all know, they are molecular machines. They are the only organelle that is seen inside a prokaryotic cytoplasm. No other organelles are present. So they are the molecular machines which are made up of, that is important point, which are made up of proteins, and there are many RNAs with specific functions inside the ribosome. So it is a protein ribonucleotide complex, or protein RNA complex is a ribosome. So ribosomal RNAs They are meant to select the tRNAs, to ensure the accurate translation, bind to translation factors, then catalyze peptide bond formation. So these are the functions of the RNAs which are present inside the ribosomes. So they select the particular tRNA, they position the particular mRNA to correct location, and they are also responsible for peptide bond formation, or they catalyze peptide bond formation. And coming to the structure, we'll see the structure in detail. So in this particular diagram which is seen below, it is nothing but it is shown, you can see both prokaryote or the prokaryotic ribosome, the two subunits are shown. Here you can see the larger subunit, and here you can see the smaller subunit, which is 30s and 50s. And inside that, so the yellow shell is the protein, and the red thing which is seen inside is the RNA. So you can see two RNAs which are present inside the larger subunit, and one RNA which is 16S. So keep in mind this particular one which is 16S rRNA, which we are going to see later, which is having an important function in initiation. 16S rRNA, it is a ribosomal RNA. So there are three ribosomal RNAs inside a prokaryotic ribosome. Coming to the sites, so these are sites are nothing but these are the regions where the tRNAs interact with the ribosome. There are three sites, and they are named according to the what tRNA does in that particular site. They are named from left to right as E, P, and A. E is for exit site, P is for peptidyl site, and A is for amino acyl site. So these are the three sites which are seen in the ribosome. And the importance of these sites is that, as you see in this particular diagram, the site is completely formed only when the smaller and larger subunit combine. In other words, if I take a ribosome, this one is the larger subunit, and you have the smaller subunit which is down, which is below. The case with the ribosome is that the larger subunit, which is the 50s, and the smaller subunit, which is 30s, they combine only during translation, or they combine only on the mRNA. Otherwise, these two subunits, the larger as well as the smaller subunit, they are seen as separate entities. They combine to form a complete ribosome only during the process of translation, and only when they combine, these three sites form completely, because 75% of the site resides in this particular larger subunit, and 25% of these sites, 25% of the spaces lies in the smaller subunit. And hence these three sites are completely formed only when both the larger and smaller subunit they combine. So as we said, from left to right, so this is the area which is called as the E site, which is followed by the next area which is called as a P site, and the next area which is the A site. So you can see a small portion of this is present in the smaller subunit also. So this is a general outline of the structure of the ribosome. And the three sites span, as I said, they span both the smaller and the larger subunit. And another thing is that the mRNA is looped into the smaller subunit, and it passes through both the sites. So taking another color for the mRNA, so the mRNA is usually looped like this, so that when the mRNA is looped into the ribosomal smaller subunit, the mRNA will pass through these three sites. The mRNA will be positioned in these three sites. It will be passing through these sites. So here the ribosome, here to be noted, the ribosome is also having a helicase activity, and always translation or the mRNA is read in this particular direction, that is from 5' to 3'. In this particular direction, the mRNA is read by the ribosome, and the anticodon portion, so the ribosome is having an helicase activity. the significance of the helicase activity is that sometimes, as we said earlier in transcription, if there are sequences in the mRNA which are complementary, that means if there are G's and C's which are close together, there are chances that in the mRNA such loops might be present. So to avoid such loops, that means mRNA should be inserted, as I showed here. mRNA should be inserted into the ribosome for translation to occur. So insertion means the particular space through which the mRNA moves, it is a perfect space only for a single stranded RNA. No secondary structures can be accommodated. As a result, if at all some secondary structures are present, to avoid that, a helicase activity is present at the particular entrance of the mRNA space. So the amino acids reside in the larger, the amino acid when it comes, it resides usually in the larger subunit. So it is nothing but, so what is said here is, so if this is the larger and this is a smaller subunit, you have the three spaces like this, and we have the clover leaf shaped tRNA. And if this is a 3' end where the amino acid, so this is the amino acid which is attached to the tRNA. Here you will have the 3 bases which will represent the anticodon. So what is said here is, when such a tRNA comes to the site in the ribosome, so for say a tRNA is here like this, so the anticodon will reach the smaller subunit here to interact with the mRNA, and you can see here the amino acid will be present in the will reside in the larger subunit. And the larger subunit, as we said, or as we saw in the diagram, you have rRNAs here, which is the 16S rRNA, and also there are 2 rRNAs here, of which one rRNA will be catalyzing the peptide bond formation. It will be catalyzing the process of the peptide bond formation. So that's with an introduction about the structure of ribosome and some terminologies which is related to translation. Now we are directly moving on to the mechanism of translation. So the first is charging of amino acid. So charging of amino acid, as we saw the picture of tRNA, it is nothing but you have a tRNA like this, a clover leaf shaped in the 3-dimensional structure. It is having a 3, because it is an RNA, it will have a 3' as well as a 5' end, and the attachment of a particular or a specific amino acid. Specific amino acid means the amino acid should be perfect. Whatever the anticodon is there, the particular the amino acid which is corresponding to this anticodon should be added to the 3' end of the 3' end of the tRNA, and here is where the amino acid is going to be added, and this particular process is catalyzed by the enzyme, which is shown here, which is aminoacyl tRNA. Aminoacyl tRNA synthetase is the enzyme. tRNA synthetase is the enzyme which catalyzes the attachment of the amino acid to the tRNA, and it is a 2-step process which requires energy, which is ATP, as seen here. ATP and amino acid join to form aminoacyl AMP, and pyrophosphate is released, that is PP, which is pyrophosphate. The aminoacyl AMP again reacts with the mRNA, so that is a specific reaction where only that is the amino acid which is corresponding to the anticodon of the tRNA will only be added to this particular tRNA, and it forms an aminoacyl tRNA. So aminoacyl usually it is written as small a. So in the further discussions, wherever we see this, please keep in mind that is it will be written like this, small a tRNA. This small a usually refers to aminoacyl tRNA. Okay, so this is the first step, which is charging of the amino acid. Charging means nothing but the attachment of the amino acid to the tRNA. So coming to the second step, we have four subdivisions in the second step, which is initiation. So in initiation, what happens is initiation and initiation complex will be formed, and now we are, as we said earlier, the ribosomal assembly should occur because the larger and smaller subunit, they are separate. It should be during initiation, these are the events that is to be carried out. That means the two subunits of the ribosome, they should combine to form a complete ribosome. Then the mRNA should be looped into the ribosome. It's not a simple looping. The looping should be perfect in the sense that the initiated tRNA should be placed at the correct site where the first tRNA will arrive. So these are the particular process that is going to happen during initiation. So we'll see the steps one by one. The first one is bringing the small ribosomal unit to the initiation codon. That is what I specifically said. Now, when the RNA is looped into the smaller subunit, it should be made sure that the initiator codon, which is AUG, should be placed at the region where the initiator tRNA will arrive. So how we'll see how this occurs. So what happens here, or how this occurs, is that so you have only the smaller subunit, the larger subunit joins together at the end of the initiation process. So during the initiation process of translation, the smaller subunit, which is the 30th subunit of the prokaryotic ribosome, first it will the mRNA will loop into the smaller subunit. So as we said, we have the three portions here, and it is at this particular P site the first tRNA with the codon, which is with the anticodon of AUG arrives. AUG is seen in the mRNA. The anticodon of AUG, the tRNA with the methionine, and will be arriving at this particular P site. So when this particular mRNA is looped, it should be noted that this particular AUG is clearly positioned in the P site. So how this occurs? So how this is correctly placed? So this is correctly placed because of a particular sequence which is present at the 5' end of every mRNA. There is a particular sequence, and that sequence is referred to as SD sequence or Shine-Dalgarno sequence, which is seen towards the 5' region of the mRNA. That is, it is seen upstream of, if we use the correct terminology, you can see this particular Shine-Dalgarno sequence upstream of the start codon. It is seen upstream of the start codon. What does this Shine-Dalgarno sequence do in placing this AUG at the correct site? So what Shine-Dalgarno sequence does is, so as we said earlier, there are many ribosomal rRNAs inside, or a single ribosomal rRNA in the smaller subunit, which is 16S rRNA. So you can see the 16S rRNA inside this particular smaller subunit. This 16S rRNA, or as we said here, your Shine-Dalgarno sequence is seen in the 5' region, or towards the upstream 5' region of the mRNA, and at the 3' region of your rRNA, and specifically the 16S rRNA in the smaller subunit, in the 3' end, there is a sequence which is complementary to the Shine-Dalgarno sequence. So what happens is, whenever an mRNA is looped into the smaller subunit, this Shine-Dalgarno sequence and the 3' end of the 16S subunit, they form complementary pairing. If a complementary pairing is formed in that particular mRNA, AUG will be correctly placed at the P site. There will be no doubt in that. if at all a Shine-Dalgarno sequence and the 3' region of the 16S rRNA base pairs, or they are complementary, as a result they will base pair. If at all that base pairing occurs correctly, it is assured that the initiation codon will be placed correctly at the P site, where P site means half of the P site, where the initiator tRNA bearing methionine will arrive after initiation. So this is how this particular process occurs. So that is what we are seeing here. So the mRNA, the ribosome assembly takes place at separate stages. It processes a specific sequence at the 5' end, which is a 5 to 10 nucleotide sequence, and that is called as the Shine-Dalgarno sequence, as we said earlier, which is complementary to the 3' end of the 16S rRNA in the 30S. So if pairing occurs, it will be correctly placed. So now coming to the other factors which are responsible for initiation, and they are nothing but the initiation factors. These initiation factors are nothing but proteins which help in translation. So now we'll see there are 3 initiation factors in the translation of prokaryotes. They are IF stands for initiation factor, that is IF. Initiation factor 1, initiation factor 2, and initiation factor 3. There are 3 factors, of which one is always associated or attached to a GTP molecule, which is an energy rich molecule. And these 3 initiation factors are attached to the 30S, or they are seen attached to the 30S ribosome, or 30S subunit of the ribosome. So taking again that 30S subunit, as we saw, our mRNA is in this direction. This is the 5', this is 3', and here if I am taking this as the 16S rRNA, so here you have the Shine-Dalgarno. This has base paired here. So this much events has occurred, and now we'll have the E site here, the P site here, and the A site here. So now we are going to see the initiation factors, the 3 initiation factors. Of the 3 initiation factors, IF1, the function of IF1 is, we'll start with IF3. The function of IF3 is found to be attached to this particular region, that is somewhat near to the E site. You can see this IF3. The main function of IF3 is to restrict the formation or the combining with the larger subunit. That means only after initiation the larger subunit should bind. So it prevents the larger subunit from joining prematurely to the 30th subunit. That is the function of IF3, which is seen towards the E site of the smaller subunit. Coming to the next one, which is IF2. As we said earlier, it is a GTP binding protein, and this is required for the attachment of the first tRNA, and this particular protein is seen at the P site. So this is a P site. It is seen towards, or so this particular IF2 GTP, this particular IF2 GTP, it is placed here. So this is IF2 along with a GTP molecule. It is seen attached to this particular site. This is A site, and you can see IF1 here. So IF1, the function. So the function of IF3 is to prevent the larger subunit from binding prematurely. Only after the initiation complex is formed, it should bind. The 2 GTP's function is it aids in the attachment of the first tRNA to the P site, and IF1 is the one, it facilitates the attachment of 30S subunit to the mRNA, and also prevent the amino-acyl tRNA from entering to the entering in a wrong position. So these are the functions of the three initiation factors, IF1, IF2 as well as IF3. So what happens is, before the larger subunit joins, the initiator tRNA joins the smaller subunits. So that is the next thing that is going to happen after the mRNA is looped, after the initiation factor join. The next is, before the larger subunit join, the first tRNA enters the smaller subunit. So the first tRNA in the case of prokaryotes, so I just draw a tRNA with an anticodon loop. So here there is the amino acid. So here you have the anticodon here at the anticodon region or the anticodon structure. So this is the initiator tRNA, the first tRNA, which is the initiator tRNA. And this initiator tRNA, in the case of, as we said earlier, it will be methionine, but in the case of prokaryotes, another formyl group is added to the amino acid and it becomes formyl methionine. So the first tRNA that enters the case of prokaryotes is a formyl methionine, because an extra formyl group is attached to the methionine only in the case of prokaryotes, and that too only in the case of the initiator methionine. All the methionine will be normal. Only the initiator amino acid methionine carries the formyl group with it. So this particular tRNA will enter the P site, because the complementary, so here you have the AUG and the complementary of AUG will be present here in the anticodon loop, and this particular tRNA will enter this particular site. And when this tRNA tries to enter this site, what happens is, whenever the tRNA is enters the P site of the ribosome, and the codon-anticodon pairing occurs. So if here you have the mRNA here and AUG is present here, and the you will have a U and A and C. AUAC will be present in the anticodon structure. They will pairing occurs, and whenever a codon-anticodon pairing occurs, two factors are released from the complex, and the two factors are nothing but our IF1 and IF3. These two factors released from the smaller subunit soon after the codon-anticodon pairing occurs. IF2 will remain along with the initiator tRNA. So after this process, you will have such a structure, that is, if this is the smaller subunit, these are the three locations, and you have the mRNA here. So what is seen is, you will have the tRNA now, which is being paired, and along with this, at this particular site, you will have your IF2 with its GTP molecule. All the other factors are now lost. Now after, when this codon-anticodon pairing occurs, IF1 and IF3 will leave the smaller subunit, and only IF2 GTP remains along with the initiator tRNA. The tRNA will be definitely having the first amino acid, which is formyl methionine. So this is the second step, which is nothing but the bringing the first amino acyl tRNA into the ribosome. Next is the third or the last step in the initiation, which is nothing but assembly of the complete initiator complex. So assembly of the initiator or initiation complex happens in such a way that whenever IF3 is removed from the smaller subunit, as we said earlier, IF3 was the one which was preventing the entry of the larger subunit. When IF3 moves out, now the larger subunit is free to bind to the smaller subunit. So definitely the larger subunit comes and binds. So when the larger subunit, that is a 50th subunit, comes and joins the smaller subunit, this joining causes the GTP on the IF2. As we said earlier, there is a GTP here. The GTP will be hydrolyzed. The GTP hydrolysis will cause a conformational change in the ribosome, and because of that so when this larger subunit is binding, GTP will be hydrolyzed to GTP, and that energy is used to produce a conformational change on the ribosome. As a result, IF2 GDP, because GTP is now used, IF2 GDP will leave the complex. So this is what is called as the initiation complex. So initiation complex will not have any initiation factor. It will have the smaller subunit. It will have the larger subunit of the ribosome, and it's a complete ribosome is now formed with an mRNA looped into it and an initiator tRNA with the formyl methionine. This particular structure is called as the initiator or the initiator complex. So the next process is elongation that we'll see in the next video.